Priority nila is that pareho silang gawa sa lipid bilayer. Okay? Pero itong mga lipid bilayer, actually marami pa yung differences. There are different kinds of lipid bilayers per cell to cell and pati na rin sa mga organelles. No? Pero yung nakikita natin sa picture, parang meron lang siyang polar head and non-polar tail. Pero yung specific name ng mga lipids na yon, okay, ay magkakaiba. Ulitin ko, magkakaiba sa bawat organelles at sa cell membrane. So on top of these lipids, magkakaiba din yung mga proteins, no? Yung mga proteins sa mga organelles na yon. Yung mga proteins na pwede nating makita sa vacuoles or sa chloroplast, sa membrane ng chloroplast ay iba sa proteins na nakikita natin sa mitochondria. No, one example of this would be yung mga photosystems. Like in this example, in this plant cell, yung mga proteins embedded dito sa chloroplast are involved in photosynthesis. Sa mitochondria naman, the proteins embedded on the surface of this organelle are associated with energy producing processes like yung electron transport chain, okay, stuff like that. So, ayan. So, yung first function or based from this figure, isa sa function ng mga membrane is compartmentalization, no? Kaya merong sariling function yung mga organelle natin, no? Dahil contain sila sa isang membrane. Second one, where's the item number 2 here? So this, yeah, yung mga membrane bound organelles, they can also serve as a scaffold for biochemical activities, like in this example, for photosynthesis. So is this yung item number 7, no? Yung membrane can also serve as yung nga, scaffold sa different biochemical activities. For the third one, ito, sa nakikita nyo, may movement yung water from outside, okay, papasok ng cell. So ano kayong function dyan? Function niya. If you are to describe this item number 3, what do you think is the function of this membrane? Just by looking at this picture. Okay, osmosis. Mm-hmm. And when we say osmosis, ano ba siya? Correct. Okay? May sumagot, selective permeable. Okay? So this membrane serves as a barrier, no? Na selective. So selectively permeable barrier. So hindi lahat kasi ay pwedeng pumasok sa cell, right? So um most of the times, kung ano lang yung essential sa kanya, siya lang yung spontaneously na pumapasok sa kanya. But yung water molecules in the lipid bilayer, it's actually very difficult para makapasok yung water molecule sa cell, especially for plant cell. Kasi, di ba, yung cell membrane, meron siyang polar head and meron siyang non-polar tail. And 'di ba, sa sandwich. So, puwedeng hindi makapasok yung water molecule because of the nonpolar component, 'no? Pero, nakakapasok naman yung water, pero hindi siya mas kabilis kapag meron tayong mga proteins embedded sa cell membrane like yung mga aquaporins. How about the fourth one? What do you think? Fourth one. ATP based from this figure. Um, hmm, ATP production or pH regulation? Well, could be. Pero maybe if this H ions is associated dito sa mitochondria, could be associated with ATP production. Pero yung membrane natin is that it facilitates the movement, no, of substances between compartments. Yes, correct. Transport. How about yung number 5? Ayan. Ano 'tong figure? May hormones dito, tapos may molecule dito na we don't know if it moved from this area, okay, papunta sa, organelle na 'to. What do you think is what could be, the function of the membrane based from this picture? Okay. Num yes, yung membrane natin, very good. It can interact with cell membrane to trigger signaling pathways. Okay, very good. For item, yeah, release signals. Okay, cell-to-cell communication, very good. Correct. Okay, how about yung item number 6? Ayan, 6. Ano to? What do you think is the function based from this picture alone? Kasi may right, may yung mga membrane meron yang continuation, right? Ano 'to? Okay. Okay, correct. Correct, Aliza. Um, yeah, plasmodesmata, the connection to other cells. So yung mga membranes natin, they also mediate recognitions and interactions sa mga adjacent cells. Okay? So how about yung item number 7? Okay, this is mentioned already. Yung energy transduction, right? So membranes transduce photosynthetic energy or can convert chemical energy into ATP. So ito, yeah, number 2 and number 7, energy transduction. So yun yung mga functions 'no ng, ating ating membrane. Hindi ko specify na cell membrane, just a membrane. Kasi when I say cell membrane, we are referring dito sa outside 'no, sa, periphery ng cytosol. Okay. So, yeah, you guys know this already, yung fluid mosaic model. No, this is how a cell membrane would look like. That's right. Yeah, I will enlarge this picture. So, and so the most common molecule that is found in the membrane is yung mga lipid bilayer, no? And yung mga lipid na to, as you see, it's made up of polar head and hydrophobic tail. Okay, so kaya nagkakaroon ng ganitong structure because spontaneous, no? Energetically favorable yung ganitong conformation. Kasi nga, these are non-polar. So as much as possible, they have to, um, ano ba, yung exposure nila sa water, dapat lesser. What else is found in this cell membrane is ito, mga proteins bound. No, yeah, you have these integral proteins. Ito, these are integral proteins. Ayan. Meron din tayong nakikitang mga peripheral proteins. We'll be discussing this in detail. And yeah, yung mga picture na to, itong mga different colors of lipids, ayan, nag-occur yan no, sa ating cell. Um, Halimbawa nito is actually hindi universal or hindi the same yung mga lipids na nakikita natin sa lahat ng ating cell. For some cells, nagsi-signal yun para kailangan na silang um, mawala sa mundo. Like halimbawa sa red blood cells, may specific kind yung mga lipids na kung saan na kapag mas marami na siya sa certain monolayer, so that will signal na sa mga macrophages para kainin na yung red blood cell kasi nag-mature na sila. As you know, nagmamature yung red blood cell and meron lang siyang certain point. So kapag mature na siya, it's readily ano na absorbable or ready na siya to be engulfed by the macrophages. So ganun yung function ng mga membrane natin. Okay, just a history. So the fluid mosaic model, so we have Jonathan Singer and Garth Nicholson. So they proposed the central dogma of not molecular biology, but the central dogma of membrane biology for more than 3 decades. It says here na it has a core lipid bilayer which exists in fluid state. Now, yung membrane natin, yung consistency niya is fluid, is an olive oil. So ganun yung consistency ng ano ng membrane or even the cell itself. So hindi siya totally solid, no? And hindi rin siya totally liquid. So ganun yung consistency ng cell membrane natin. And membrane proteins form a mosaic of particles penetrating the lipids. So, yan. At this point of time pa lang, I want you guys to acquaint yourself na sa different parts or different biomolecules associated sa cell membrane. So, tandaan nyo no, na yung mga integral proteins, meron silang membrane spanning domain. So, ibig sabihin, meron silang region na meron na parallel sa non-polar tail ng lipids and meron din silang polar regions na makikita dito facing extracellular and facing intracellular. Pero hindi lahat ng proteins ay integral or meron siyang membrane spanning domains. Some of them are weakly associated lang, no? And we call them peripheral proteins. The bonding between these proteins, this integral protein, is or could be a very weak ionic interaction. And ano pa yung nakikita natin sa picture na to? And yung protein na to, some of them are alpha helix, pero though common yung alpha helix sa cell membrane, pero meron ding nag-occur na mga beta structure tulad ng mga porins nga. Kasi this beta structure can form barrels. So, associated din sa lipid bilayer natin is yung mga cholesterol. We will discuss this in detail mamaya. What else is in here? Meron ding mga sugar moieties like a short um, fragment ng oligosaccharide. No? So we know na kapag mas marami yung ratio or mas marami yung protein compared sa carbohydrate, we call that glycoproteins. Pero kapag mas marami yung sugar compared sa protein, we call that proteoglycans. Okay? Now, yung sugar molecules, hindi lang siya bound sa mga proteins, pwede rin siyang mag-bind sa mga lipids. And, okay, so yung mga bonding between this protein and yung carbohydrate, madidiscuss naman mamaya, meron tayong tinatawag na N-link and O-link glycosylation. Okay. So, ayun no. So, based from this picture, nakikita natin na very complex yung ating cell membrane. Hindi lang siya gawa sa lipid bilayer. No? And kung lipid bilayer man yan, meron pa yang diversity. So, meron yang, based from this, no, meron siyang lipids, meron siyang phospholipids, meron siyang cholesterol, may proteins. Kapag yung proteins bound sa carbohydrate, we call that a glycoprotein. Or if yung lipid bound sa sugar, we call that a glycolipid. Okay. So this is how a cell membrane would look like. This is a stalk of membrane. So ito yung cross section ng ng neuron. Ito yung myelin sheath, no? And this is an axon. So yung myelin sheath na yan, 'di ba? As you know, it serves as an insulator and para mas mabilis yung pag-jump ng information, no? Yun yung importance ng myelin sheath. It serves as an insulator para mabilis yung pag-transmit ng information. And ayan, pwede nating makita yung structure niya via the electron microscopy. So we already know this na lipids and proteins are the major components no of your membrane. And on top of these two biomolecules, meron din tayong mga carbohydrates. Nabanggit ko na rin 'to sa inyo na lipids are amphipathic, meaning meron siyang polar and non-polar, and meron tayo 'to, um, variants ng ating membrane lipids. We have phosphoglycerides, we have lipids, and ito cholesterol. So, and sa phosphoglycerides pa, marami pa 'to. Pero we'll just focus lang sa major definition of what phosphoglycerides are. These are, they have diacylglycerides with small functional heads linked to glycerol backbone and phosphate ester bonds. Actually, pwede nyo naman siyang ma-context clues based from the name itself. No? Yeah, let's say, no, there is this phosphate ester bonds, as the name suggests, no, there's this phospho. Okay, so yung glycerides, so ibig sabihin, meron tayong glycerol, no? So sphingolipids naman are ceramides formed by the attachment of sphingosine to fatty acids. We have ample examples for sphingolipids. And last is yung cholesterol. They are smaller and less amphipathic lipid that is only found in animals. But may mga nagki-claim na meron din daw cholesterol in plants, but some would say that it's not really cholesterol but cholesterol-like lang sila because cholesterol are strictly found in animals. So this is a comparison between a lipid and a cholesterol. So ayan, ganito rin yung orientation niya sa ating cell membrane. So kung yung lipid, no, let's say this is facing this is the non-cytosolic and ito sa baba yung cytosolic. So kung if this is cytosolic, so ang facing sa cytosolic monolayer, okay, is yung mga polar. Okay? So yung mga polar ends. So yung orientation ng cholesterol ganun din. Kung nasaan yung polar head, andun din yung OH, yung polar region ng cholesterol. So yung non-polar tail ng phospholipid, ganun din yung orientation ng cholesterol. This is the non-polar, yung flattened carbon rings. Okay? And we'll be discussing in a while ano ba yung function ng cholesterol in terms of fluidity ng cell. Okay. So this is a liposome. Okay. So kapag meron tayong mixture ng individual um, lipids, okay, kapag meron tayong mixture ng phospholipids, kapag nilagay natin siya and in-incubate siya sa water solvent, spontaneously magkakaroon ng ganitong structure, no? Magkakaroon ng ganitong structure. For some, they call it micelles, okay? Pero this is a synthetic vesicles. Okay. So, ano ba yung importance ng mga synthetic vesicles in genetic engineering? This is one way para sa drug delivery. No, halimbawa, yeah, in this figure, meron tayong drug, okay, contain kasi let's say no, hindi intravenous, let's say yung drug na yon ay in an experimental animal, yung route niya is via oral gavage. Yung drug. So, eventually, kapag bare lang, kapag naked lang yung drug, kapag taken orally siya, it will interact sa ating mga sa mga proteases or yung mga enzymes that would digest proteins. No? Especially if protein-based yung inyong drug. So, and pagpasok niyan sa intestine, makaka-interact niya yung strong acid, no? So that that acid might degrade or destroy or denature yung inyong target proteins or molecules. So one way to protect our drug is to enclose it. So and one of the easiest way is to do a synthetic vesicle. So in this example, ayan, so aside from the lipids, meron ding bound antibodies. Bakit merong antibodies dito? Kasi 'di ba kapag nag-take ka ng drug, let's say the drug has to be specific, yung action niya has to be specific in your liver. So papaano 'yon? Or let's say the drug has to be specific sa inyong small intestine, let's say. So one way to do it is to couple it with an antibody kasi the antibody is very specific to the antigens or to whatever receptors that is found in the target organ. No? So let's say no sa small intestine merong receptor doon. Um yeah, kasi hindi naman lahat ng cells ay the same yung receptor, right? Kaya nga sa COVID-19, isa sa manifestation bakit nagkakaroon ng or isa sa manifestation ng COVID-19 is diarrhea. Kasi we have a receptor for that virus in our intestine. Pero yung receptor na yon might not be found in certain parts of our body. Okay? And using that concept, yung antigen and antibody interaction that can be utilized in designing your vesicle. So, pwede rin naman na um, yung drug mo ay especially no, let's say yung drug mo is non-polar, it is non-polar. So yeah, you can protect it kasi it can incorporate itself sa non-soluble segment or region of that lipid bilayer. So more likely this is um a protein that is hydrophilic. So ito yung protein. If this drug is a protein, definitely this is hydrophobic. Kasi if this one is hydrophobic, what happens is they will just tend to cluster together. Magkakaroon lang ng aggregation. So, yan. And yeah, in this example, they design this vesicle with the use of this PEG or polyethylene glycol. Um, yung PEG kasi is very actually common to sa mga makeup sa mga makeup enthusiasts diyan na kasi it prevents yung para yung it preserves yung moisture or yung hydration sa mga makeup kits. And sa vesicles naman or sa mga synthetic vesicles, ginagamit yung PEG um, for um, paano ba natin explain siya? Para hindi siya ma-recognize ng ating immune system to be foreign. Okay? So, yeah, gumagamit ng PEG for that. Yeah, to escape the immune system, tagging ng ating immune system. Okay, so it says here, lipid bilayers assemble spontaneously in aqueous solutions as in the form of a liposome. Okay. And okay. In this figure, eto, ayan. So these are your lipids. Oh, you have the head and you have the tail. And magkakaiba 'no, yung, actually yung mga blood types, yung O antigen, A antigen, or blood type B, they differ based sa attachment ng carbohydrates 'no, sa surface ng cell. So for O antigen, it has this molecular structure. It has a glucose, galactose, and acetyl glucosamine. Ayan, may fucose, galactose. Ang difference niya naman sa I-antigen is bound sa galactose is and acetyl galactosamine. Ayan. So meron din tayong two types 'no na kung saan ang protein pwede siyang ma-conjugate or mag-bind sa carbohydrate. Ito yung common, ayan. So this is this is your amino acid asparagine, and this one naman is the serine or threonine. So itong curve-like lines na 'to, these are polypeptide backbones or can be a peptide bond. Um, Ayan. So puwedeng mag-bind. So yung bonding ng sugar sa protein ay mediated by end base and glycosylation. Puwedeng end glycosylation, okay, if ang nag-interact dito is yung amino group, okay, yung nitrogen containing compound of that amino acid. So, yeah. For some naman, okay, the interaction naman is not in the end, but in oxygen. So, yeah, so this one is N-linked glycosylation, and this is O-linked glycosylation. So for N-linked glycosylation, this is displayed by the amino acid asparagine. But for O-linked glycosylation, yan, yung mga amino acids involved in here are serine and threonine. Okay? So, yan. So this is an acetylglucosamine. And sa baba naman, when you have this serine or threonine, this is an acetyl galactosamine. Ayan. So, let's say, ayan, ang difference ng, ng A antigen sa B antigen is that ito sa terminal end, for A antigen, it has an acetyl galactosamine. So ibig sabihin, the amino acid bound or that mediates the interaction is either serine or threonine. Okay? So for B antigen naman, it's not the GalNAc, okay, but just a regular monomer galactose. So, ayan. I hope that is clear na meron tayong two types of linkages that join sugar to polypeptide chain. The N-linked glycosylation and the O-linked glycosylation. Okay. So, yep, basics lang naman to. This can be achieved by reading. Glycolipids have larger carbohydrate chains that may be cell-to-cell recognition sites. Carbohydrates of the glycolipid of RBC's plasma membrane determines the person's blood type, as explained. So, okay. So we'll discuss now yung mga different proteins embedded sa ating membrane. Though nabanggit na siya kanina, yeah, so one example of this or one kind of membrane protein is the integral protein. Pwede siyang, it occurs, pwedeng isa lang, okay, or pwede siyang chains, no? So in this example, this is just a, let's say, in its tertiary level, meron siyang one alpha helix membrane spanning domain. Okay, so for this protein. And for this second integral protein, meron siyang 1, 2, 3 alpha helix membrane spanning domain. Okay? So, okay. So ulitin natin, no? I think we discussed this last time na the amino acid residues facing this region should be or must be hydrophilic. So is in this region. Pero yung mga regions facing these non-polar regions are definitely non-polar amino acids. Okay? So ibig sabihin ba nito lahat ng mga amino acids na makikita sa membrane spanning domain ay non-polar? Well, hindi naman lahat. Okay? Let's say, and hindi rin lahat, though common yung mga alpha helix, hindi lahat ng mga membrane spanning or ng mga integral proteins are in alpha helix conformation. Some of them can be in the beta structure. So this is how a beta barrel integral protein would look like. So yeah. So itong interaction nito, ayan, so this is, so the protein runs anti-parallel. Diba, we said last time na yung beta structure can run parallel or anti-parallel. And the bonding that mediates this bonding is yung hydrogen bonds, right? It's the hydrogen bonds between the amino and the carboxyl. Okay, so the carboxyl and amino of the other. So, yan. So itong structure na to, itong barrel na to, one example of this nga is yung tulad ng na-mention ko kanina, is yung mga aquaporins. Okay? Is yung mga aquaporins kasi yung mga aquaporins, ayan, eh, very easy lang yung movement ng water para makapasok yung water outside, papasok sa inside of the cell. And if we do a cross section of this lateral side ng beta barrel na to, ayan, makikita nyo na merong yung mga R groups, merong non-polar. So ibig sabihin the one facing this lipid bilayer, ito yung mga non-polar regions are cubic R groups. Okay? Or yung mga non-polar R groups. Pero yung nakikita naman sa loob, since para siyang barrel, so yung mga R groups naman na makikita doon ay mga polar. Okay, mga polar amino acid residues. So, if I'm gonna ask you true or false, lahat ba ng mga acids na meron or parallel sa non-polar tail ng lipids ay non-polar din? So, the answer is false, no? Kasi it depends sa structure and hindi siya nag-occur sa lahat ng proteins. Okay. So we're done with the integral proteins. So ulitin natin no na yeah, this is an example, itong beta barrel na to. This is an example of a channel proteins. Okay. So, ang tawag lang sa segment na to, ayan, basta yung nag-e-extend na to, these are called the membrane spanning domain. Okay? The membrane spanning domain. Okay. So they can serve as a receptor, a transporter like an aquaporine, or an agents that transfer electrons, yung mga yeah, yung mga proteins involved sa electron transport chain. Okay, next is we have, so by the way, no? So in here, walang walang interaction or hindi covalent yung bonding ng mga ng protein na to sa part na to ng lipids, no? Okay, so ganito lang yung interaction niya because this is more energetically favorable for them. Kaya spontaneously ganito yung kanyang conformation. So hindi covalent, no, yung bonding ng R groups na makikita sa protein na to and sa lipid tail. Okay. So meron din tayo mga peripheral membrane proteins. So they are just a weak type of bonding and they are normally or they are commonly hydrophilic. Okay? They are commonly hydrophilic na nagpo-protrude sa bilayer. And since very weak lang yung kanyang interaction, so just by changing the pH of the cell, readily na sila na ma-remove. So very weak lang yung interaction ng mga peripheral membranes. So they are located entirely outside of bilayer on either the extracellular or the intracellular. Okay. Or tinatawag natin na ito. So if this is the cytosol, itong baba, kung ito ang cytosol, okay, and this is definitely extracellular, ang tawag sa monolayer ng lipid na to ay cytosolic monolayer. Okay? So the one that is facing the cytosol is called the cytosolic monolayer. And yung other side niya naman, this is called the non-cytosolic monolayer. Okay? Ulitin natin. The one facing the cytosol or yung cytoplasm is the cytosolic monolayer, and the one facing the extracellular component is the non-cytosolic monolayer. Okay. So non-covalent yung type of bonding. Yeah, they serve as a mechanical support. They can also act as an enzyme. May mga proteins kasi embedded sa membrane na kung saan they can also act as a catalyst. Halimbawa, no, when we are to absorb disaccharides and bago siya ma-absorb sa ating cell, dapat kailangang i-cleave or ma-hydrolyze yung disaccharide into a monosaccharide. And to do that, we need enzymes. And some of those enzymes are embedded sa surface ng cell membrane, especially sa mga cells found in our intestines kung saan nangyayari yung absorption process. Ayan. There's also lipid-anchored membrane proteins. Okay. So as seen in this figure, ayan. So meron tayong this is phosphatidylinositol. Ito yung lipid. Okay, coupled yung ating lipid or yung protein. This is our protein, no? So this is the GPI anchored protein. Okay. And meron dito mga phosphate, the mannose, the N-acetyl glucosamine, the inositol phosphate, and the lipid. So the bonding between this protein and this lipid is done via covalent bonding. So very strong, no? Very strong yung pag-incorporate ni GPI anchored protein na to sa ating lipid. For some, yung mga Lipid-anchored protein naman, okay, so yung incorporation niya is, weak interaction lang. Okay, so one example yung GPI, ma-encounter natin yung mga 'to, yung mga GPI, as we proceed na sa cell-to-cell communication. Kasi, when we go to cell communication, we will be encountering a lot of proteins. So paulit-ulit siyang babanggitin like yung mga GPCR or yung mga G-protein coupled receptor, eto this GPI or this, glycophosphatidylcholine inositol. Okay. So so we have three. Ulitin natin yung nauna. We have, this is an integral protein. Okay, yung peripheral protein is weak interaction lang siya sa integral proteins. Right? Or weak interaction lang siya, okay, um sa lipid head. Okay. So more likely mga, zwitter ion lang yung interaction niya, mga weak interaction lang. What else? Ayan, so membrane lipids exist in gel. So yung consistency niya, right? It's like an olive oil. Exist in gel or liquid crystal phases depending on, these are the factors: temperature, lipid composition, and saturation in the presence of a cholesterol. Blah, 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 at warm temperature. Sige, let's try to explain this na lang. Okay, cholesterol interact. Sige. Ayan, let's try to explain this na lang, through this figure. Okay, so we have figure A and figure B. Okay, sa chat box, though it's very apparent, between A and B, which do you think is more solid? A or B? Marami nagkakamali dito. Oh, B, 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 B. Okay, may sumagot ng A, B, A, B, B, A, A, A. Okay, nagiging dumadami na yung A. Okay, syempre charot lang. It's very obvious, no? That the figure B is more solid kasi they're more intact. So yung movement dito sa B is constricted compared to the figure A. Okay. So in the chat box, bakit kaya more solid ang B? Bakit kaya ganito yung structure niya versus sa A? Ano kayang difference nila in terms of the structure? Bakit more spaces yung meron sa A compared sa figure B? In the chat box, go ahead. Okay. Parallel structure gives more stability, I think. More compact. Yes, yeah, this is more compact. It's very apparent naman that this is more compact. This is more solid. But why, do you think? Mas konti yung space sa B? Yes, it is. And it's because of what? Because of the fluid content? Okay. Okay, so, all right. Some answered temperature. Temperature. Okay. Lack of water content. Okay. Um, Sige. Wait lang. Minimize ko lang yung chat box. Ayan. So sa figure na to, yeah, you guys are correct. At least most of you. This one is more solid and this one is more liquid. Let's say you expose them at the same temperature. Let's say 100 degrees Celsius, or no, not, that's very extreme. Maybe at around 45 degrees Celsius or 42, let's say 42. So medyo kung nilalagnat ka ng 42, oh, delikado na yon. So at 42 degrees Celsius, what do you think will happen? Okay. At physiological pH, sa chat box, I want you guys to respond. At 42 degrees Celsius, what do you think will happen to your cell membrane? Will it be more liquid or more solid? No one's responding yet. At higher temperature, what do you think is happening? Or let's say you have yeah, you're suffering from colds and nagkaroon ka ng lagnat. Okay. So, at least apat, lima, anim, pito. Okay. So far, out of 10 respondents, 9 responded na they tends to be more liquid, which is true, no? Kapag just think na lang, yung example nito is yung mga mantika. 'Di ba? Kapag nilagay mo yung mantika sa higher temperature, what happens? Tends to become what? They tend to become more liquid, right? Pero when you decrease the temperature, kapag binababaan natin yung temperature, nagiging more solid siya, right? So, ganun din yung ating cell, no? Na kapag mainit, nagiging ano siya, mas more movements, no? So, halimbawa, no, let's say ako, let's say hell is real. Hell is real. Nasa impyerno ako kunyari. Um, Syempre patay na ako noon. Pero syempre, hypothetical lang naman 'to. Um, yeah. So nasa mainit ako na lugar. So yung mga cells ko, no, matutunaw yung mga 'yan, matutunaw. Dahil yung mga cell membrane ko, they tend to be more fluid. So magme-melt talaga siya, no? And let's say gusto ko pang mabuhay, gusto ko pang maka-survive, gusto ko pang mag-exist. So ang gagawin ng cell ko, dahil nagiging more fluid na siya, sobrang fluid na siya, so ang gagawin ng cell ko ay ang gagawin niya ay kailangan niyang pahabain yung kaniyang tail. Okay? So yun yung gagawin ng cell ko para maging more solid siya dahil nagiging liquid na siya eh. So kailangan maging solid siya. And para maging solid siya, different ways. Una, yeah, pwedeng mag-add ng hydrocarbon chains, longer hydrocarbon chains sa aking lipids para maging more solid siya. And pangalawa is in this figure, no, may mga kinks. And as we said last time, may mga kinks yung lipids dahil sa mga double bonds, no? So these are unsaturated fatty acids. So yung mga unsaturated fatty acids, sila yung may mga double bonds, sila yung may mga kinks, sila yung may mga bands, sila yung more fluid, more liquid. And ayoko yun kasi nagiging liquid na yung ano ko eh, nagiging liquid na yung cell membrane ko. Diba? I have to cope up with this temperature, with this very high temperature. So, what my body needs is ayan, I have to elongate my lipid tail. Pahabain ko yung hydrocarbon chains ko and I have to remove those double bonds. I have to convert or my cells have to convert those unsaturated fatty acids into saturated fatty acids para maging more solid siya kahit meron ako sa high temperature. Okay? So, that's one way kung bakit, ayan, yeah, kaya sabi dito, kaya hindi uniform, no, yung ating membrane sa magkakaibang temperature and in different conditions. So, it says here, structure depends on the temperature. So, Could be na ang ginawa ng cell ko dahil sobrang init, inutusan niya yung aking DNA na mag-produce ng mga enzymes na involved sa pag-convert ng unsaturated fatty acids into saturated fatty acids. Okay? Or kung baliktad naman, let's say, um, pumunta kayo sa sobrang lamig na place. Okay, sorry. See. And pumunta kayo sa sobrang lamig na place, no? So, ang mangyayari is, yung mga membranes nyo, titigas yan, right? Magiging more solid yan. And ayaw natin ng solid kasi kung solid ang ating cell, di ba, what will happen? Hindi magkakaroon ng movements. No? Walang movements. And mamamatay tayo kapag walang movements sa cell. So, dapat, it has to be fluid. So, since parang think of of the lard. LARD, yung mga mantika na mas nasa malamig na, let's say pag December, di ba? Or sa amin lang siguro. Kapag malamig, di ba, nangyari sa mantika, tumitigas. So kung cell yon, ayaw niya ng matigas. Gusto niya fluid lang. So baliktad yung gagawin niya, no? So ang gagawin niya ay puputulin niya yung mga chains, yung mga fatty acids na to para maging more fluid siya. So yun. So kanina 'di ba, pinapahaba yung hydrocarbon chains. Pero kapag malamig naman, no, gagawin niya yung the other way around. Paiiksiin niya yung kanyang tail. What else? Pararamihin niya para magkaroon ng spaces, no? Um, para magkaroon ng spaces. So anong gagawin niya? Dadamihan niya ngayon yung lipids na may mga unsaturated tails. Okay? Para more kinks, more movements, more fluid. Okay? So, yeah. So that's why structure depends on the temperature. So the fluidity of membranes is a compromise between structural rigidity and complete fluidity. Membrane fluidity makes it possible for proteins to move in the membrane and for membranes to assemble and grow. Kasi, ayun nga, kapag let's say may mga proteins dito, no? And yung mga proteins, kailangan nilang mag-move kasi. Nag-move ang proteins. No, hindi sila stuck dito. Nag-move sila laterally. Ayan, I think I have an example for that. And yung movement na yon, may mga function yon, bakit sila nag-move. Though, hindi lahat ng proteins nag-move. Kasi may mga stuck sa coral. And maintaining membrane fluidity. Organisms other than birds and mammals maintain membrane fluidity as temperature changes by altering the composition of the membrane to be cold resistant, and fatty acyl chain single bands are desaturated, forming double bands. Chains are reshuffled to produce two unsaturated chains. Ayan. Reshuffling ng phospholipases na nagka-cut ng fatty acids. What else? Ayan. So, yep. So, familiar na tayo, no? Sa term na structure depends on the temperature. Let's now go to the next. Ayan. And yung ating lipids, yung ating lipid bilayer, ano yan? Asymmetric yan. Hindi equal yung distribution sa non-cytosolic and sa cytosolic monolayer. And yung mga lipids na to, nagmo-move din yan. Nagmo-move yan. Yeah. So, let's try to check. So, yung mga movement ng mga lipids na to can be in lateral or in transverse. Lateral, let's say from this side, okay, going to this side. Pero it's not gonna jump. no? Parang wag niyo isipin na parang, mag-jump to. No, it's not jump. It's just a lateral movement. Or this cytosolic monolayer, the cytosolic monolayer lipid can move to the non-cytosolic monolayer. Pwede rin yun. And also, ayan, nagmo-move din yan. Yung since the bonding between the lipid head and the tail is single bond. And di ba nabanggit ko sa inyo na single bonds, they are freely rotating. Okay? So this one can rotate as well. Ito, clockwise or counterclockwise. And yeah, the fastest so far is this kind of movement, the flexing, lateral followed by lateral shift. And it takes time, no? You need more time para sa transverse diffusion. And ayan, yung mga movements na to, ayan, ano to, this spontaneous to. Hindi mo kailangan ng enzyme para makapag-rotate to, para makamove siya. And even this lateral shift, you don't need energy para makapag-shift siya. But, ayan, if it's transverse diffusion, if there is a transfer between or the movement from cytosolic to the non-cytosolic or the other way around, we need ATP. And not only ATP, we also need enzymes like the flippase, the floppase, and scramblase. So for flippase, you're moving from the non-cytosolic to the cytosolic monolayer. If it's floppase naman, from the cytosolic monolayer to the non-cytosolic monolayer. So yan yung mga enzymes involved. And this requires energy, adenosine triphosphate. And to balance things out, so so this flippase and floppase increases asymmetry o yung differences, no, ng cytosolic and non-cytosolic monolayer. Yung scramblase, so yung ginagawa niya naman is it randomly um it randomly moves the monolayer from cytosolic to non-cytosolic. So it decreases asymmetry naman yung scramblase. Okay? Random yung movement nito. And in the red blood cells, yung scramblase, they are actually inactive. But when you increase yung calcium, ang ginagawa kapag mataas yung calcium is that nagiging active. Okay? Nagiging active si scramblase wherein yung phosphatidylserine, okay? So ang ginagawa niya is from the non-cytosolic monolayer, yung mga phosphatidylserine na meron sa cytosolic monolayer, tinatransfer niya sa non-cytosolic monolayer dito sa taas na to. And kapag marami ng phosphatidylserine sa non-cytosolic monolayer, remember na itong part na to ng lipids, ito yung nakaharap. Ito yung facing sa mga immune cells natin, sa mga macrophages. Okay? So kapag recognize ng mga macrophages na maraming phosphatidylserine sa red blood cell na to, that's the time na kailangan niya ng i-engulf si red blood cell. Ibig sabihin, due na siya. Ready na siya to be discarded. Okay? So, ayun yung dynamics ng plasma membrane natin. Okay, this would only show you yung diffusion. This is how they show diffusion in yung proteins embedded on the cell membrane. So what they did was they fused the human cell and the mouse cell using the addition of Sendai or the fusing virus, no? So, okay, so yung mga hollow circle na to, these are protein from the human cell. And this dense protein na to, densely colored, these are proteins sa mouse cell. Okay, so when they fuse, ayan, when they fuse, as you see, um, there is a concentrated protein sa human cell and sa mouse cell. And this can be best visualized using this. Ayan. So may mga red and may mga blue. Ibig sabihin, ongoing yung merging ng mouse cell and ng human cell. And this coloration was done because of, ayan, because of ELISA. So what they did was they simply incorporated primary antibodies specific to the proteins found in human and proteins found in the mouse. Okay? So yung mga mouse ay green labeled, fluorescent, and yung mga proteins naman for human are red labeled. So they observe through time. Kung after fusing ba, what do you think will happen? Magmo-move ba to? Magdi-diffuse ba siya equally? So after 40 minutes, ayan. So nagkaroon nga ng movement, no? Yung mga proteins natin. So randomly, some of them scattered na yung red, no? So hindi na wala ng concentration, wala ng gradient yung yung protein yung proteins from the mouse and from the human. No, it's equally distributed na. But is this true for all? Na lahat ba ng proteins ganito ba yung mangyayari kapag ganito ba yung nangyayari sa cell? Well, the answer to that is no. Kasi there are some proteins, ito, the one highlighted in pink, these proteins restrict the movement of this. Let's say this is an integral protein. So they call it a coral. No? So kaya if hindi man nag-move, could be attributed yon sa mga proteins forming this coral. Okay. And ayan. And meron ding difference aside from that, meron ding differences yung proteins found in different locations. Let's say in the intestine. So this is the cross section of your intestine. Yung space nito, this is called, ano ba yung space sa inyong gastrointestinal tract? That is called the lumen, right? The space, lumen. So the proteins facing the proteins that are found facing the lumen is different from the one facing the blood vessel. So this one kasi, this is how the glucose molecule is being absorbed in the cell. And ayan, this is a co-transport. Ayan, so this is ang tawag nga ulit dito sa mga microvilli. Yeah, sorry, microvilli. Yeah, so these epithelial cells, sa surface nila, yan, nandiyan yung mga integral proteins. No? These integral proteins, bakit ko sinabing integral to? Bakit hindi ko binanggit na peripheral protein siya? Well, the fact na kailangang mag-cross ng glucose from out to inside of the cell. So ibig sabihin, there has to be the presence of the transmembrane, the membrane spanning domain, right? So kaya ko siya sinabing integral protein. So is this. This has to be an integral protein. Now, yung glucose molecule, right? When absorbed, not when absorbed. The movement of the glucose dito is, hindi ko rin pwedeng sabihin na porque kumain kayo from high concentration to low concentration. Kasi, hindi naman yung concentration ng glucose sa cell natin ay always less. Kino-convert lang niya yung glucose into parang less like by um by putting phosphates on it, no? Kapag phosphorylated yung glucose, kapag phosphorylated yung glucose sa cell natin, yan, hindi niya na-recognize na it's a glucose. Okay, kaya lesser yung concentration, 'di ba? Kaya napapansin niyo sa glycolysis, what happens? Meron yung mga phosphofructokinase, yung mga kinases, right? Kailangan pinophosphorylate yung mga glucose molecule. Kasi kapag hindi mo siya pinophosphorylate, like glucose lang siya, so yung movement ng glucose is not spontaneous. Dapat kasi ang movement is from high concentration to low concentration. No? So kung glucose tapos non-phosphorylated glucose, medyo mahirap yung movement nun. No? Hindi spontaneous yung movement nun. So dapat sa loob ng cell, dapat hindi siya glucose conformation. Dapat it's not glucose, pero glucose. So it has to be phosphorylated. No? Kaya ang nangyayari, ang akala ng cell, kulang yung, kakaunti lang yung glucose niya kasi, ayun, it's phosphorylated. Yun nga, it's a glucose na may phosphate groups. So, in here, yeah, the glucose use yung sodium ions kasi there's a gradient no sa lumen outside ng cell and inside of cell. So, and parang ano siya, parang sa sa pag-produce ng electricity, 'di ba yung turbine? So, nabanggit ko sa inyo last time na hindi iikot ang turbine kung pareho lang yung level ng tubig sa dam versus tubig sa river, right? Hindi magmo-move yun, hindi spontaneous yung movement ng water, no? So dapat from high concentration to low concentration. And medyo user si glucose kasi ginagamit niya yung opportunity na yon, ginagamit niya yung concentration ni sodium para makapasok siya sa cell. Okay? And ayan, another integral protein, yung disaccharide. Kaya sabi ko kanina, right? From disaccharide into monosaccharide. So, ano bang process yun? Hydrolysis, right? Hydrolysis, when you cleave or when you cut this disaccharide into a single monomer. And that can be acted upon by the enzyme that is incorporated in the cell membrane. Kaya sabi natin kanina na isa sa mga function ng enzyme, ng integral proteins or ng protein is they can also act as an enzyme. So ito yung function na yun. No? Now, so you have a different proteins that is found in the apical part of the cell, and you have a different protein naman that is found sa basal region facing the blood vessels kasi from the glucose, sorry. Ayan. So from the intestine, papasok na yan sa ano, papasok na yan sa um, what do you call this? Sa blood vessels, specifically sa mga capillaries. Okay? So iba yung protein na makikita sa apical, sa basal, and sa lateral regions. Okay? So the one that is found here naman, meron ding mga, dito na papasok yung mga tight junctions. Ayan, itong mga tight junctions na to. Yeah, halimbawa nito is yung sa ano, sa paano ba example? Good example natin for this. Sa spermatogenesis and sa oogenesis. 'Di ba? Yung teka lang, paano ba natin ano to? Okay. Okay. So if this is if this is a testis, ayan, yung mga Sertoli cells, ayan, kung saan nagmamature yung mga sperm cell, 'di ba? The one that is found dito sa basal region If the lumen is yung seminiferous tubule sa testis, 'di ba kapag nag-cross section tayo, ito yung space dito dito yung dito yung mga sperm cell, dito sila magta-travel kapag mature na sila. Kung testis 'to, ayan, so saan ba nakikita yung mga hilaw na mga sperm, 'di ba, sa basal region dito sa part na 'to. So dito, and as they undergo meiosis, magmo-move sila from basal pababa sa lumen. Kapag spermatozoa na sila, kapag mature sperm na sila, ano mangyayari? Lalabas na sila, no? They will just so yeah, they will just dissociate themselves from the cell, papasok dito sa lumen, and from the lumen magta-travel na sila sa seminiferous tubule and travel outside of the body after ejaculation. So, um, yun, may mga proteins involved doon. Yung mga tight junctions, no? Na sila yung, ang function nila is yung blood testis barrier. No? So, yung mga sperm cell, hindi sila nakasurvive, hindi sila nakadeposit in between cells. Yung mga sperm cell, nagmamature sila sa loob mismo ng cell. Okay? And yung mga tight junction na to, these are said to be blood testis barrier kasi yung mga sperm cell, kailan ba sila na-produce, di ba? During adolescence. Di ba? Kung kailan ka nag-mature, saka lang mapaproduce yung sperm cell. And since late siyang na-produce ng ating katawan, it's being recognized as foreign by our immune system. No? So let's say this is the blood vessel. Kapag andito yung mga macrophages natin, or yung mga immune cells, 'di ba? So without this blood brain sorry, not blood brain, blood testis barrier. I'm sorry. Yung blood testis barrier. So without this junctions na to, pwedeng maka-penetrate, makapasok yung mga immune cells, and they will engulf, no, yung mga sperm cell kasi nga they recognize sperm cells as foreign already. Okay? So, kaya yan, may mga tight junctions, may mga different proteins na yung location nila ay nakadepende, okay, sa kanyang function. Okay, I hope that is clear. And yeah, is another example is yung sa sperm cell. Okay? Kasi you need you need proteins dito sa acrosome. If the sperm is made of the head, the midpiece, and the tail, sa head meron siyang acrosome, nandun yung not digestive enzymes, pero just like digestive enzymes that will digest the zonal layer of the egg cell. And dapat, and so bakit yung fertilization ay nangyayari between or intra-species lang, not inter, no? So that is because of the specific proteins na makikita sa surface ng head ng sperm cell, like the ZP binding molecule, na recognizable lang siya sa mga ZP proteins or yung mga proteins na makikita sa surface ng zona pellucida. Okay? So yeah, and those ZP proteins are concentrated or can only be found in this part of the cell. Okay? So yes, so you also have different proteins that is found on the surface of the tail. Ayan. So may mga proteins din na specific sa midpiece. And in some studies, those proteins would signal this egg cell para i-reject yung midpiece. And as you know, yung midpiece nandoon yung ating mitochondria for male, no? And yung mitochondria, andun yung paternal DNA, no, mitochondrial DNA. Kaya 'di ba, yung mga tayo, wala tayong mga yung mitochondrial DNA natin ay galing lang sa nanay kasi nga during fertilization, no, walang nasasamang mitochondria sa male. So kaya nga yung pagtatalo na sino ba ang mas maraming ambag sa anak, no? Is it the mother or the father? Kasi sabi nila equal naman daw kasi 23 chromosomes from the mom and another 23 chromosomes from the dad. Pero technically, it's incorrect kasi mas marami tayong DNA na na-inherit sa ating nanay because of the mitochondria. Even as you know, yung mitochondria, it has a DNA on its own. Di ba? Meron tayong mitochondrial DNA. So, kaya mas maraming ambag sa atin, nanay or tatay, it's always the nanay, genetically speaking. Okay. So, and share ko lang din no na when I work sa Philippine Carabao Center, when we assess the sperm cell, kasi aside from the concentration ng sperm cell, chine-check din namin yung kanyang morphology. Kung ready ba siyang magbuntis or not. So chine-check din namin yung number of tails. Some of the sperm cell, like multi-tailed sila. Yung iba naman, walang head. Yung iba, sobrang laki ng head. And yeah, isa rin sa chine-check namin is yung acrosome integrity. Acrosome integrity. Kasi kapag sira na yung acrosome, like let's say a lot of sperm cells found in the semen, and very low na yung integrity ng kanyang acrosome, hindi na siya makakabuntis, no? Dahil wala siyang enzyme to digest the layer of the egg cell. And ano pang ginagawa namin na? Okay. We also evaluate yung membrane integrity. And we do this by staining using safranin. Yeah, it's safranin. So kapag um kapag colorful or yung intensity ng color sa cell ay sobrang taas, ibig sabihin patay na siya. Kasi nga, kapag patay na siya, wala na yung integrity ng kanyang cell membrane. Na-absorb niya na yung mga stains. No? So kapag living pa siya, kapag viable pa yung mga sperm cells, ibig sabihin, hindi niya na-absorb yung mga stains namin. So that is how we do viability assay. Is through staining. Kapag na-absorb yung stain, ibig sabihin, sira na yung cell membrane, nakapenetrate na yung stain. Pero kapag na-reject yung stain, ibig sabihin, intact pa yung cell. Ibig sabihin, viable pa siya. But don't get confused this with staining ng mga bacteria sa gram-positive and gram-negative, right? Iba yung mechanism nun. It has nothing to do with the integrity of the cell membrane, right? Ang ini-stain mo lang naman doon is yung peptidoglycan, no? Yung thickness. So, ayan. Next. So, movements of substance across cell membrane. Meron tayong dalawa dito. We have the active and the passive transport. Kapag sinabi nating passive transport, we don't need ATP to do its action. Kapag active transport, we need ATP. Okay? So, under passive, meron tayong non-mediated and transporter. Sa active, pwede ring transporter and mediated. So, sorry. Ito, halimbawa, for, ayan. So these three are passive transport. Kapag passive, ibig sabihin, spontaneous yung reaction. Energetically favorable siya kasi it follows yung movement from high concentration to low concentration gradient. Okay? So, like the oxygen, ang oxygen, they can freely cross the membrane kasi um, hindi siya ionized kasi oxygen or let's go back doon sa explanation ko nung una, 'no? Kasi merong non-polar dito, right? May non-polar dito. Itong tail na 'to non-polar. So ang oxygen has to be non-polar. Is it non-polar? Let's try to go back doon sa electronegativity difference. Kapag dalawa yung oxygen, let's say halimbawa lang, ang electronegativity ng isang oxygen ay 1, the other oxygen is 1. 1 minus 1 is 0. So there's no electronegativity difference. So ibig sabihin, if walang electronegativity difference, it is non-polar, right? So ayan, that's why it can freely cross the cell membrane. For water molecule, medyo may difficulty siya kasi nga ayun nga um meron siyang partial positive and partial negative. Though it can cross naman, pero hindi siya kasing efficient kapag meron tayong integral proteins like the aquaporins. Yung mga ions, yan, yung mga ions, hindi yan nakakapag-cross basta-basta sa membrane. These has to be regulated. And yeah, especially mga bigger molecules like the glucose, they need integral proteins. They need a transport protein. So, ayan. So dito, ayan, from high concentration to low concentration. Pero bakit hindi kailangan ng ATP when you transport glucose? Diba nabanggit ko kanina na it utilizes yung gradient ng sodium ions, right? So ginagamit niya yung energy, yung potential energy na meron sa sodium ion para makapasok siya sa loob ng cell. And ayan. And also, hindi kailangan ng ATP para ma-absorb yung glucose molecule, no? Hindi kailangan ng ATP to do that. Kasi nga 'di ba, nauubusan ka na nga. Naub let's say wala ka na ngang energy, kulang ka na nga ng ATP, kumain ka. Tapos gagastos ka pa ng energy para ma-absorb mo yung glucose, 'di ba? Hindi siya very efficient. So, ayan. Kasi nga kailangan nga natin ng ATP coming from the glucose molecule. So another active transport is kapag sabing active transport, we require ATP. So ATP is hydrolyzed into ADP and inorganic phosphate. And one example of this is yung transport ng sodium ion and potassium ions. And this is an antiport. Antiport kapag opposite yung direction ng movement ng ions. Uniport kapag single lang siya. For glucose, this is co-transport or symport kasi you also need sodium ions para makapasok si glucose molecule. This one is uniport lang to. Ayan. So, yung sodium potassium na to, example nito is yung sa neurons, right? As you know, nagte-travel yung information because of this potential difference, yung potential difference inside and outside of the cell, no? Yung movement ng sodium and potassium. Kaya nagte-travel yung information because of this, the sodium and potassium, and involved dito yung sodium potassium pump. And sa movement na to, kailangan natin ng ATP. No, that's why just by staring at this slide, so yung information from your optic nerves na mag-travel yan papunta sa inyong utak, and sa pag-travel ng mga ions na to inside and outside of the cell, ayan, nag-spend na tayo, gumagastos na tayo ng energy. No, yung ATP is hydrolyzed into ADP. Okay, simple diffusion through the lipid bilayer. Ayan, facilitated diffusion via the protein transporter. Okay, so yung mga non-mediated, yung mga diffusion lang to. Okay. So yung mga uncharged inorganic, these can freely move. Yung oxygen, carbon dioxide, okay, nitric oxide, water less kasi water are polar and meron tayong non-polar ends, no, ng lipids. Pero they can still cross. So just a review of your movements ng water. So yan. So let's say no, stuck ka sa island and uhaw na uhaw ka, 'di ba? We know na hindi mo pwedeng inumin yung salt water para not just to relinquish your thirst, pero yeah, kasi instead na ma-hydrate ka, made-dehydrate ka pa, right? Because when you drink salt water, it has a lot of solutes. It has a lot of sodium chloride. So kapag when you have more sodium chloride, what happens? Ang gagawin is yung cell mo, um, yeah, ang gagawin ng cell mo is lalabas pa yung mga water molecules. Kasi ang gagawin niya, i-dissolve niya, no? I-dissolve niya yung surroundings niya na recognize niya as a lot of solute. So kapag when you have more solute, what happens? Water tends to move out of the cell, which will make the cell shrink or crenate, no? So this is the condition of the cell under hypertonic solution. If it's isotonic solution, yan, when you have an equal concentration of solute outside and inside of the cell, so yung movement ng water is equal lang. No, there is no net movement of water. So for us, for animals, this is more favorable for us, isotonic solution. But for plants, they don't want this, no? What they want is yung hypotonic solution. So, ang kailangan ng plants is that less yung solute outside, kaya diligan mo lang sila, no? Kasi um if hindi hypotonic yung solution, what will happen? Magiging flaccid. Magiging flaccid lang yung cell. Flaccid is parang malambot, like if you left your yung fries nyo overnight, di ba nagiging flaccid siya, or yung ano ba yun? Yung tite kapag lumambot, no, it's flaccid. So, ganun. Sa plants to maintain yung turgor pressure kasi 'di ba, dapat yung water ma-store sila sa vacuoles and the force in the vacuole will push outward doon sa cell wall. So, kaya for plants, yan, favorable sa kanila yung hypotonic solution. But for animals, no, we don't want hypotonic solution. No, it has to be an isotonic solution kasi let's say you are a doctor and yung organ kailangan temporarily store mo siya. You don't need a pure water, no? You don't want to submerge your organ in a pure water kasi what will happen, the water will just be absorbed by your cell in that organ and all cells are all gonna burst. And we don't want that. So, yeah, so that's why, yeah, for us, this is more favorable for us, the isotonic solution. No? So kung ano man yung condition sa physiology sa ating ay normal physiology sa human body, so yun yung kailangan mong i-mimic, no, sa lab. Okay, diffusion, osmosis, hypotonic, and this is what I'm talking about. Yung for plants, it's called plasmolysis, no? Plasmolysis. Kapag nagkakaroon ng ayan, kapag hypertonic yung solution kasi yun nga yung sa vacuole, no? So, for this, we will continue this next meeting. Yeah, next meeting na lang 'to kasi it's already 7:53 na. Ayan, so do you guys have questions? Okay, I swear okay, thank you for reminding me. Okay, i-upload ko na siya. This slide and yung biomolecules. May question kayo, guys? Or comments? Or may na-gets ba kayo? Mas naintindihan niyo na ba yung cell membrane? Yes or no? At least. Okay, thank you for validating for okay, okay. Ayan, so for the other class, wala na tayong meeting tomorrow. And so next week na tayo. So I will be uploading the slide bago ako umuwi kasi I'm still in the office. Okay, so do you guys have questions? Or suggestions or comments? Go lang. May questions ba? Comments? Masyado bang mabilis? Or what? Okay. So, okay. Yes, po-post ko na, I swear, yung previews. Though, sabi ko parang, it's in the ano naman, it's in the book, pero, you know, yung may mga slides din kasi ako na hindi ko kinuha dun sa book na in-upload ko. Yeah, information overload. Ano pangalan tayo? Sorry, pang 20 pa lang 'to, 20 slides pa lang 'to. Pero yes, yeah, information overload talaga ang sell and mall. Pero lahat kasi ng explanations, some ang daming applications ng mall bio, no? Sa medicine, kung paano ba yung mechanism ng yung pathology, everything, what was going on in the lab, no? So that's why we have to understand talaga yung molecular biology kasi ayaw nating maging mukhang tanga in the future kapag maglalab na tayo, we don't know the basic concepts. And ayun din yung ayokong, kasi it happened to me, like ayoko mangyari sa iba, ayoko mangyari sa inyo. So, yeah. Kasi it's okay na information overload kayo ngayon kasi you guys, understandable na information overload kasi nasa stage kayo na kailangan nyo munang i-absorb lahat ng information kasi there's no other way, no? Or yeah, you can read pero hindi naman tayo lahat we have the same drive para magbasa. No? And sometimes ang difference is that kapag nagbasa ka, ang problem nun is hindi mo alam kung saan ba dito yung magiging useful sa akin in the future. No? So hindi mo alam kung ano yung kailangan mong alamin talaga kasi ang dami-daming mga words na feeling mo hindi siya magiging relevant. So yeah, that is one disadvantage din na kapag um kaya ayoko rin na parang, "Oh, please read this." Ganyan, bibigyan nila ng read, "Next meeting, we'll have an exam." Kasi I don't know, maybe it's not for me, it's not working on me na bibigyan lang ako ng reading materials tapos in-expect na alam ko na. Ayun, kasi nangyari yan sa akin, like yung oh my God, yung professor ko nagbigay siya ng reading ano, siyempre last week, reading break sa UP. Reading break pero nagbigay ng ano, ng babasahin. Ayun, nagbigay siya ng mga kailangan naming basahin and ayun, wala lang, sobrang hirap lang tapos yesterday we had an exam. My God. Nagsisisi ako bakit ko siya in-enroll. And I thought magaling na ako sa pag-process ng whole genome. Ayan. Pero, well, actually, ang ginawa namin is, yung exam namin, basahin niyo lang 'to, tapos bigla kami nag-practical exam sa whole genome. Tapos medyo, oh my God, sana pumasa ako. Pero kinakabahan ako. Tapos nag-asa ako kanina dito sa RITM. Ang kinaganda naman dito sa job ko ngayon is that we do have a lot of bioinformaticians. And I just learned na yung ginagawa namin is totally different from what they're doing. No, not totally different, pero just because nagsi-sequence sila, they do process sequences, eh the same na yung mga codes, commands, yung mga software, sobrang iba din pala. So I just realized na ano, ang daming kailangang aralin at hindi mag-e-end sa BS yung mga inaaral. Ayan. So the more you know, the more you realize na medyo syunga-syunga ka pa talaga. Anyways. So yun lang. It's okay guys no na ma-information overload kayo kasi nasa stage pa lang talaga kayo ng pag-absorb ng maraming information. Mahirap na at this stage na limit na yung mga nakuha niyong info, mas kawawa kayo in the future. So that's gonna be all. Thank you very much for attending and enduring this ilang oras tayo, 6, 7, 8. Okay, salamat and bye-bye.